Hello everyone. It's a great pleasure for me to present my research work at OHBM 2021, and I'd like to thank the organizer Dr. Jing and Dr. Vince Cowan for the kind invitation. In this presentation, I will introduce two fMRI studies that show why learning from high-dimensional data is important for pain neuroscience and how this concept can be applied in terms of neuroimaging. Among the many different types of human perception and cognition, pain is one of the most highly distributed processes in the brain. For example, in the seminal study of functional brain mapping led by Dr. Penfield, patients rarely reported pain in response to the numerous focal stimulation of the brain. This suggests that activation of a single neuron or a brain region may not fully represent the experience of pain. Also, neuroimaging researchers have reported the activation of bilateral distributed brain regions in response to experimental and clinical pain. And lesions of these brain regions, such as bilateral damage to entire insula or cingulate cortices, did not fully eliminate the ability to perceive pain. Here is an illustration of brain circuit for pain processing. Overall, these findings suggest that pain is processed in a highly distributed and degenerate manner, and may explain why the previous studies to find the pain center of the brain have failed. Therefore, to understand how pain is processed in the human brain, it is crucial to learn the core low-dimensional representation from the complex high-dimensional neural data that contain distributed and degenerate information of pain. In this regard, I'd like to introduce two of our studies that aim to learn pain from high-dimensional neuroimaging data. First one is about developing brain-based biomarker for sustained pain, entitled as Tonic Pain Signature or TOPS. And second one is about examining dynamic brain representations of sustained pain based on network science approach. Here's the first study. Developing pain biomarker has been a topic of great interest because of its neuroscientific and clinical implications. And recent advances in multivariate pattern analysis allowed pain researchers to model the quantitative relationship between the high-dimensional neuroimaging data and subjective pain ratings. Here are the representative examples of fMRI-based biomarker for pain. These biomarkers show generalizability across multiple datasets that are not used for the model training, suggesting that it is possible to extract low-dimensional representation of pain from high-dimensional neuroimaging data. However, most of the previous biomarkers, including these examples, were derived from the experimentally induced brief basic pain, such as thermal pain, lasting less than a minute. Although most of the clinically prevalent and important pain disorders usually sustain over a long time, more than a month. This temporal characteristics of clinical pain is thought to be an underlying reason for well-known differences between experimental basic pain and clinical pain. For example, in terms of their perceptual quality or the underlying neurobiological mechanisms. In this study, we targeted the sustained nature of pain and aimed to develop a brain-based biomarker for sustained pain using whole brain functional connectivity. We experimentally induced sustained pain in the lab. In other words, experimental tonic pain, with the hypothesis that the brain representations of the experimental tonic pain can be generalized to clinical pain. To induce tonic pain with the required level of safety and effectiveness, we developed a new protocol using capsaicin-rich habanero hot sauce or capsaicin concentrate. Capsaicin is a major ingredient of hot chili pepper and activates the TRPV1 channel that transmits the nociceptive signal to the ascending pain pathway. In our experiment, we delivered the capsaicin-rich hot sauce onto the participant's tongue using a filter paper, inducing an orofacial tonic pain. Then, we recorded the participants' brain activity while they were experiencing painful sensation in their mouths for about 10 to 20 minutes. Here's an overview of our experiment and analysis. In study 1, while the participants were experiencing tonic orofacial pain, we scanned the participant's brain and then calculate functional connectivity, and then develop multiple machine learning models to track the dynamic changes of tonic pain ratings using different combinations of features and algorithms. Then, the best model was selected based on a predefined criteria, including sensitivity, specificity, and generalizability across the Study 1 training dataset and Study 2 validation dataset, obtained using similar experimental design. The final model was tested on a total of 4 independent test datasets. 2 datasets, including Study 3 and supplementary data, were tonic pain datasets, and the other 2 datasets, including Study 4 and Study 5, were clinical back pain datasets. Here is the final model, named Tonic Pain Signature or TOPS, trained using principal component regression techniques to learn the low-dimensional representation of pain from high-dimensional connectivity space. Colors represent the predictive rate of each region-by-region connection. When we examine the network-level pattern of the predictive rates by averaging and thresholding, we found that most of the strong positive or negative predictive rates were between network connections, not the within network connections. Anatomical region-level pattern of predictive rates also show that between network connections had strong predictive rates. These findings support the idea that neural representations of tonic pain have distributed and degenerate characteristics. We tested the tonic pain biomarker on the tonic pain datasets. The top panels represent the test results from validation dataset that is used for model selection but not for model development. And the bottom panels represent the test results from independent dataset that was collected from different sites and not used for model selection and development at all. First, in the sensitivity test, we examine whether the TOPS can predict individual fluctuation of pain ratings within tonic pain experience. Each line in the left plot shows the within individual correlation between actual pain and signature responses, and 2 color traces in the right plot represent the group level average of actual pain ratings and signature responses across time. The TOPS tracked 10 to 20 minutes of individual sustained pain experience with reasonable performances. The right bi-only plot shows the overall signature responses within each experimental conditions, including capsaicin, and also bitter taste or aversive odor that can cause tonic aversive but non-painful experience. The TOPS successfully discriminated capsaicin condition from tonic aversive condition. Overall, this suggests that the TOPS is a sensitive and specific biomarker for tonic pain. In addition, these are the results from supplementary data. There is an additional, totally independent unseen dataset. In this experiment, participants were delivered the capsaicin or bitter tastant twice throughout the run using an MR-compatible ghostometer system. Results show that the TOPS tracked the ratings with a significant correlation and showed 2 peaks at the time of the maximum pain. In addition, the TOPS didn't respond to the bitter tastant again, demonstrating its specificity. Then, to examine whether the tonic pain biomarker can be generalized to clinical settings, we tested the biomarker onto clinical back pain datasets. These datasets were obtained from a public data repository, openpain.org. The TOPS show significant prediction for the individual differences in overall pain severity of patients with subacute and chronic back pain, although the performance depended on the types of experimental task. For the subacute back pain patients, the model worked while the patient paid attention to their spontaneous pain, but for the chronic back pain patients, the model showed medium to large effect size for both spontaneous pain rating task and also resting time. In addition, the TOPS showed significant classification accuracy for discriminating chronic back pain patients from healthy controls in 2 additional open datasets. Here's an interim summary. We developed a biomarker for tonic pain based on whole brain functional connectivity and demonstrate that neural representation of tonic pain is highly distributed across distinct functional networks. The biomarker named as TOPS showed high sensitivity and specificity across multiple independent test datasets. And the TOPS could also predict overall severity of clinical back pain and also discriminate patients with back pain from healthy controls. Overall, the TOPS successfully captures the core sustained component of pain that is shared across experimental and clinical conditions, suggesting its potential for clinical translation. However, This biomarker study focused on the predictive performances and between individual generalizability, so detailed description about how neural representations dynamically changes during sustained pain experience within individual is lacking. It is well known that pain is not static, rather a dynamic experience. For example, there is a continuous fluctuation of spontaneous pain in patients with chronic pain disorders, and pain is significantly affected and modulated by dynamic changes of attention and mind wandering. From this motivation, we conducted a follow-up study to examine the dynamic change of brain representations during sustained pain. Network science is a great tool for capturing low-dimensional, useful information from high-dimensional dynamic connectome data. And here we used multi-layer community detection method that is specifically designed to capture time-varying changes of community structure of a dynamic network. This dataset is the study 3 of the previous tonic pain biomarker study. So each participant was scanned for around 20 minutes with and without capsaicin delivery, while continuously rating their pain in the scanner using trackball device. Using the multi-layer community detection, we aim to examine the difference of brain community structures between resting state and sustained pain and the dynamic change of brain community structures over time. Here's an overview of analysis. First, we calculated dynamic connectivity matrices using 2-minute non-overlapping moving windows and thresholded the matrices based on network density 0.5. Then, we used multi-layer community detection method and then converted the community assignments into module allegiance values that was defined as 1 if 2 regions are assigned to a same community and 0 otherwise. This module allegiance values of each individual more than averaged to infer the group-level consensus community structure. Here are the results from consensus community detection. Although the community structures of capsaicin and control conditions look similar, there are notable differences in dorsolateral prefrontal cortices and also subcortical regions including thalamus and basal ganglia. Overall, the red-colored community 2 was enlarged in capsaicin condition compared to the control condition, and distributional profile of canonical networks show that the integration of frontoparietal and subcortical regions with the somatomotor dominant community 2 in the capsaicin condition was the main driver of this change. This enlargement of somatomotor dominant community is in line with the concept of pain supersystem from a previous study of Jing and his colleagues. Lastly, we examined the consensus community pattern of early, middle, and late period of sustained pain that correspond to the first 6 minutes, next 8 minutes, and the last 6 minutes. Although the community structures for each period look similar, differences can be found in dorsolateral prefrontal cortices and also subcortical and brainstem regions. Overall, the red-colored community 2 was big in the early period of pain and then gradually decreased, but the sky blue colored community 6 was initially small but gradually enlarged in the late period of pain. And distributional profile of canonical network show that the decrease of frontoparietal regions in community 2 and the increase of frontoparietal regions in community 6 were the main driver of this dynamic change. This suggests that in the early period of pain, somatomotor dominant community is integrated with frontoparietal regions to form the enlarged pain network, but as time goes by, the frontoparietal regions are integrated with each other to form their own community and may serve as an independent top-down regulatory network. Also, the purple-colored community 4 was enlarged and indigo-colored community 5 was decreased in the late period of pain, and this is mainly driven by subcortical and brainstem regions that change their affiliation from community 5 to community 4 in the late period of pain. This suggests that integration of cerebellum with the subcortex and brainstem is important for pain regulation in the late period. Here's the summary of the second study. We demonstrate that brain community structures are not static and dynamically reconfigured during sustained pain. And during sustained pain, frontoparietal regions were integrated with somatomotor dominant community to form pain supersystem. And as pain is diminished, frontoparietal regions were separated from the somatomotor dominant community and formed their own community. and subcortical and brainstem regions were integrated with cerebellum dominant community. Overall, these results show that brain circuits outside of classical pain processing pathway, such as frontoparietal and cerebellar regions, contributed to the construction of sustained pain, and therefore emphasized the distributed and degenerate mechanisms of pain processing again. We went through two fMRI studies that aim to learn the neural representation of pain using machine learning and network analysis from the high-dimensional neuroimaging data that contain distributed and degenerate information of pain. Thank you for your attention, and I'd like to thank my amazing mentor Wani, and also all the co-authors for their great contributions. Here's our lab photo. Thank you.
Hello, welcome to my presentation. I'm Qunfang from the MLSP lab at UMBC. I'm very pleased to be here to share our work, the subspace analysis of multi-subject fMRI data that enables the subgroup identification, which then can help study the heterogeneity of neurological disorders such as schizophrenia. Due to the dynamic nature of human brain, we also investigated the use of dynamic features in subgroup identification. There are three parts in my talk. First is a brief introduction to the background of our work, and then followed by two parts of our work on subgroup identification, which enables us to study the heterogeneity of disorders. In the introduction, I'm going to talk about the data-driven methods and the subspace analysis of fMRI. Data-driven blind source separation techniques, such as independent component analysis, ICA, have a bunch of advantages and have been widely used in analyzing fMRI data in order to extract meaningful imaging features. In our study, the analysis of multi-subject fMRI data, we used independent vector analysis IVA that extends ICA to multiple datasets by making effective use of the dependence across datasets. IVA is able to preserve subject variability by forming source component vectors that collect corresponding components from all the datasets. This makes IVA a promising method for subspace analysis. When applying IVA to subspace analysis, we extract three types of subspaces. The common subspace that consists of SCVs with components that are all highly correlated. In this subspace, the information is shared across all subjects. The distinct subspaces consist of SCVs with components that are almost not correlated. In this subspace, the information are unique for individual subjects. The subspace of group-specific information consists of SCVs with components that are highly correlated within subgroups of subjects. These subgroups can be identified using proper post-analysis. With large amount of effort, we are getting better understanding of neurological disorders such as schizophrenia. The study of their subtypes can help make better decisions in personalized treatment. Will the identified subgroups indicate schizophrenia subtypes that can help us study the heterogeneity? Next, I'm going to talk about our work that gave a positive answer to this interesting question. In the first part of our work, we addressed the computational challenge of IVA, proposed a framework IVA for common subspace analysis, and studied the heterogeneity of schizophrenia using static features. When applied to multi-subject fMRI data, IVA suffers from dimensionality issue, which says that the performance of IVA decreases when the number of latent sources and data size increase beyond a range. This is because the number of samples, that is the number of voxels in fMRI data, is fixed. The number of latent sources can be reliably estimated using existing algorithms. We then designed a hybrid simulation to investigate the influence of the number of data sites in our case when using the corporal resting state fMRI data. The simulation results suggest that 50 is the best value when IVA yielded the best performance. We used a subset analysis that enables us to use this selected value as the number of data sites in a given IVA decomposition. We developed a framework IVA for common subspace that consists of two stages. In the first stage of subset analysis, we randomly divided the dataset into subsets and applied IVAs to two individual subsets. And we get 3 rough subspaces. In the second stage of common subspace identification, we finalized the 3 subspaces using the pipeline shown here. By further analyzing the group-specific SCVs, we are able to identify subgroups. This is the pipeline for subgroup identification. The left figure demonstrates the 3 subspaces. The common subspace consists of SCVs with high correlation values, and the distinct subspaces consist of SCVs with low correlation values. In the middle, we have the SCVs with a mix of high and low correlation values. The components that have high correlation values are shared within subgroups of subjects. To identify the subgroups, we clustered the SCVs by using their correlation matrices as features and modularized the mean correlation matrix in each cluster. From the modularized correlation matrix, we see clear subgroup structure. We found brain network components that demonstrated significantly higher or lower activation in different brain regions for a certain subgroup compared with the other subgroups. More importantly, we found that these identified subgroups demonstrated significant differences in terms of their clinical pain symptoms that are measured by pain scores. By looking into their individual symptoms, we found that different subgroups possess different unique symptoms. For example, as shown here, the subgroup 1, it has a unique positive symptom and a unique negative symptom, while the subgroup 3 shown here has multiple unique general symptoms. The results tells us that the identified subgroups are potentially associated with schizophrenia subtypes with different clinical symptoms. In the second part of our work, we studied the dynamic features, the activity, connectivity, coevolution patterns, and studied the heterogeneity of schizophrenia using these dynamic features. We applied sliding windows to the obtained data and extracted dynamic features in both temporal and spatial domains. We applied adaptively constrained IVA to extract the variabilities from the window datasets. AC-IVA incorporates reference signals into IVA. The use of reference signals reduces the effect of dimensionality issue and eliminates the alignment problem in subset analysis. In our work, we used the common static components as the reference signal. The use of their spatial maps as the reference signal provides an efficient quantification of the dynamic changes in spatial domain. We quantified the spatial property of the dynamic boat activity by measuring the correlation between the estimates and the reference signals, and quantified the temporal property of the dynamic boat activity using 12 features. By investigating the cross-correlation between them, we found that the two types of dynamic boat activity tend to have synchronized patterns. And there are more components demonstrating synchronized patterns for the healthy control group compared with the patient group. There's evidence that both boat activity and functional connectivity are related to the mental and the cognitive processes in human brain. It would be desirable to study their coevolution patterns in order to get a better understanding of dynamic patterns. We coordinate the dynamic boat activity and the dynamic functional network connectivity to yield coevolution matrices. By studying the coevolution matrices, we found two components that yielded significant coevolution patterns for the healthy controls, but only one component for the patient group. And this is the component that merges multiple brain regions. For this component, there are more significant coevolution patterns for the patient group compared with the healthy control group. The preference of this complex component that merges multiple brain regions may indicate the abnormally organized brain for the patients. We identified subgroups using these dynamic features, and the different values are used as the number of subgroups. As shown here, in the three cases, all the identified subgroups demonstrated significant differences in terms of their dynamic patterns. And we found the subject clusters that are consistently detected across the three cases, as shown in different colors and the matrices. More importantly, when there are 6 subgroups are identified, the subgroups demonstrated significant differences in terms of their clinical symptoms that are measured by PANSS scores. By looking into different categories of PANSS scores, we found a patient subgroup that is centered on the default mode network, a network that has been reported in many studies to be associated with unique functional patterns in patients with schizophrenia. This subgroup demonstrated negative correlation between their negative and positive PANSS scores, and it is centered on the DMN network, DMN component, because this component is significantly correlated with multiple other functional network components. To summarize, the results of our work demonstrated that IVA is an effective algorithm for subspace analysis, and both the static and dynamic features are promising in subgroup identification. Neuroimaging data is a potentially important tool for studying the disorder heterogeneity. This is a list of our published papers in case that some of you are interested in getting more details of our work. At last, I would like to thank all the team members from the MLSP lab directed by Professor Tule Adali for their great help and support during my work. Our work was supported in part by the NSF grants and NIH grants listed here. Thank you.
To you today very quickly about multi-frame evolving dynamic functional network connectivity states, EVO DFNCs. This is a method for representing fluidly varying patterns of functional integration in the dynamic connectome. This talk will be very short, and for those intrigued and interested in learning more, I would suggest having a look at our bioRxiv preprint with the title given at the bottom of this slide, and we'll mention it again at the end of the talk. So the typical pipeline for investigating shorter timescale dynamic functional network connectivity, at least within a certain corridor of the research community, looks a lot like this. Basically, you decompose the brain using either group ICA or some sort of atlas or a seed-based method into a set of functional units. You then take the time courses of those functional, in our case, functional networks, which represent the contribution of that functional network to the global signal at each point in time. You pairwise correlate the time courses of the functional networks and do this on, typically we used to do this for all time points in the scan, and we would get what we now call a static FNC matrix. And nowadays we are also taking that same approach, but we're using it on shorter windows in the scan, and we're computing the pairwise correlation between all the network time courses for successive sliding windows through the multivariate time series. This results in a bit of a data explosion. We now have about usually over 100 high-dimensional DFNC observations or windowed FNC observations per subject. And the first pass at kind of managing this data explosion and learning something from it was to approach it with basically something like a hidden Markov paradigm. And so what's done is usually the observations are clustered in a time-blind way, so just all of them together, although each has a time index, the time indexing is ignored, and the observations are just thrown in as just a collection of samples, and they're clustered. The centroids of the clusters are treated as the states of the hidden Markov model, and the dynamics is characterized by mean dwell times within clusters and transition probabilities between clusters. So why is this hidden Markov model simplification so limiting for brain dynamics? It's obviously somewhat limiting for probably any number of complicated and poorly understood systems, but in resting state fMRI, it's unusually limiting and potentially consequential. So in resting state fMRI, the connectome is smoothly varying. It does not exhibit the abrupt phase transitions of, for example, condensed matter physics. This smoothness arises from biological constraints, from the constraints of bold imaging as a modality, and is further consolidated by the smoothing effect of sliding window correlations. Furthermore, the brain supports behavioral and cognitive processes that are highly branched and contingent. The Markov assumption is particularly limiting for this kind of memory-laden dynamics. And quickly, I'll have you just look at the schematic on the lower right, which shows a history-dependent transition probabilities with a non-forgetful router state that determines what comes next based on longer chunks of the incoming itinerary. The state immediately preceding the entrance to the non-forgetful router state is the same for both of the incoming trajectories. So that state is clearly holding more information if it is functioning in the way is shown in the schematic, where it takes the itinerary on the bottom left and produces the outgoing trajectory on the upper right, and does the itinerary on the bottom right and goes to the outgoing trajectory on the top left. And the histogram on the left is simply something that shows some of the consequences of handling these smoothly varying processes in Euclidean space as though they consisted of meaningful discrete states. So this just shows the ratio of the distance of every observation to the centroid of the cluster it belongs to, divided by the distance of that observation to the next closest centroid. And the mean of this distribution is 0.98. It's a very narrow distribution, and it just kind of indicates that most observations are not meaningfully closer to the centroid of the cluster they're assigned to than to some other centroid in the system. So arguably, we believe this suggests that data-driven methods for capturing group-level representations of fluidly evolving time-resolved connectivity will support more effective investigations of the dynamic connectome. Ideally, these representations would evolve in a naturalistic manner, which is to say they would evolve smoothly. They would capture distinct evolving patterns rather than multiple phase shifts or magnitude shadows of one or two dominant patterns. They present interpretable explanatory contributions to observe DFNC sequences, and they would be easily tunable for different timescales. So there are a number of challenges to this project, and I won't go into them here, but our approach has been to basically embed the high-dimensional dynamics in the plane in such a way that the key features of the high-dimensional group-level and subject-level dynamics are preserved in the embedding, and then study the dynamics in the planar embedding kind of via the differential geometry of the planar embedding, and lift those findings back up into high-dimensional space. So the embedding algorithm that we use is uniform manifold approximation and projection, UMAP. It's well suited for this particular kind of project and is less stochastic, for example, than t-SNE, though it's not 100% stable. And so what we do is we tune, there's two key parameters of UMAP. We tune them in such a way that we will end up with individual subject trajectories in the embedding that are tightly continuous and such that the group level geometry in the plane is tight and entangled, as this is reflective of what you see in the high dimensional case. Just to be very clear, the average distance between successive observations within subject at the high dimensional, in the high dimensional space the mean element-wise distance between successive observations is 0.006. They're really tightly continuous in general. And moreover, the individuals, if you take an observation from a given subject in the high dimensional space and look at the mean element-wise square distance from its 10,000 nearest neighbors that are not observations from the same subjects, we're not subject to that continuity constraint for within subject trajectories, the mean element-wise distance between that observation and its 10,000 nearest neighbors is less than 0.01. So these trajectories occupy a very tight bundled space in the high dimensional state space, and we want that entangled geometry to be reflected in the embedding as well. So what we do, first we do 25 of these embeddings, and just to stabilize, we then average those and use the averaged embedding, which you see in the bottom, the second from the left on the bottom. And in that case, you'll also notice that although we retain the individual subject continuity, these individual curves that you see are subject trajectories. So they're the embeddings of individual subject trajectories. So the trajectories embed as kind of continuous curves in the plane. But although they're very entangled in the individual UMAP embeddings on the left, they're a little bit sparser after averaging. So the next step is to approximate successive intervals on each embedded curve with a line segment, characterizing each with its midpoint and slope. And then we create summary linearized 2D exemplars by clustering all of the segment midpoints and slopes. The centroids of this clustering are triples, xk, yk, mk. xk, yk are coordinates, mk is the slope. Each cluster centroid summarizes the position and direction of a subset of the 2D segments into which UMAP embeds higher dimensional DFNC trajectories. Corresponding to each of the clusters, we construct a linearized 2D DFNC trajectory exemplar, gamma k, of slope mk, centered at xk, yk. Gamma k has geometric length Lk, which is based on a distribution of segment lengths in cluster K. It is sampled at tau evenly spaced points along its length, and so it has temporal duration tau. Tau is fixed by us, and that's a parameter that we chose. So for us, tau is 44, which is twice the window length of the window used in the computation of the DFNC observations that are inputs to this model. Okay, to build some intuition, I'll just show you this set of visuals here. So we're zooming in. We have the embedding in the top left, a square in the embedding that you see. We zoom into that square, and then on the top right, what you see is the field of linearizations, the tangent segments that we compute along each of the curves within that box. Those are the objects that are being clustered. Each of them has a midpoint and has a slope, and also has a length. And so the midpoints and slopes are clustered, and the length of the trajectory exemplar for that cluster is determined by the average length of the linearization, the local linearization in that cluster. And then you see again at the bottom right, superimposed on the embedding, a set of linear exemplars. Okay, we build high-dimensional EVO DFNCs from the 2D linear trajectory exemplars in the following way. As mentioned before, each of the exemplars is divided into tau evenly spaced points. The EVO DFNC corresponding to linear exemplar K, or let's say, for example, the linear exemplar in the middle of this figure, is determined by we lift each successive point of these evenly spaced points up to a frame in the EVO DFNC. And we do this by taking the average of the inverse images of the 25 nearest planar neighbors to that point in the exemplar. So that point has 25 neighbors that are actually in the embedding and have true inverse images. We average those inverse images, and then we produce that frame of the EVO DFNC. And we do this at every point along the exemplar to create the full EVO DFNC associated with that exemplar. You'll note that we are showing, although tau for us is 44 TRs, we're showing much shorter. These are subsampled versions, just because it's already a lot, and so we decided to subsample for display. Okay, so the next step is to actually use these EVO DFNCs as basis objects, basically dynamic basis objects, with which to represent actual windows of length tau from observed subject DFNC sequences. And as you can see in the bottom figure with the with the linear exemplars distributed over the embedding. Not all of the subjects, represented by curves in that embedding, are necessarily close to a given, are necessarily very close to a given linear exemplar, and so we actually go ahead and use them as basis objects to reconstruct the observed windows within subjects, and this creates a multivariate time series where we have weightings for each window for each EVO DFNC. So you see here, subject 266, 81st DFNC window of length tau is a weighted combination of these EVO DFNCs on the top right, and we do that at every window and create this multivariate time series representing the full sequence of DFNCs for every subject in terms of the EVO DFNC basis objects. So you can see here that there are pervasive effects of schizophrenia on the representational importance of the 10 EVO DFNCs we computed for this study. In solid green boxes are EVO DFNCs that have significantly greater representational importance in controls. In red boxes are EVO DFNCs that have significantly greater representational importance in patients, in both cases after correction for multiple comparisons. And there's a number of kind of interesting EVO DFNCs here, and we will skip any descriptive narrative here just in the interest of time. Another interesting feature of the EVO DFNCs is that they carry fairly strong effects of positive symptoms of schizophrenia, such as hallucinations, delusions, paranoia, grandiosity. Unlike the hidden Markov model states from this particular data set, which has been studied rather a lot, we do see some strong effects of kind of the positive psychotic symptoms of schizophrenia on the representational importance of these EVO DFNCs. Quick kind of way of looking at a quick example of why embedding some of these patterns in longer itineraries or longer evolving motifs is important is you see, we'll call this pattern DMN neg. This is the negative DMN to the rest of the brain connectivity. We see a high contrast version of this in EVO DFNC2, followed by weak connectivity. In EVO DFNC3, we see a persistent but low contrast version of this DMN neg pattern, and in EVO DFNC7, we see a medium contrast version followed by hyperconnectivity. EVO DFNCs 2 and 3 are both more important in schizophrenia patients. EVO DFNC7 features the same pattern, but embedded differently in its evolving motif, and that one is more representationally important in controls. It's also the case that in terms of positive symptoms of schizophrenia, EVO DFNC3 carries the greatest number of these significant effects, and it's the least dynamic, and in a way we would not really be able to see the importance of this low contrast, persistent, relatively non-dynamic DMN neg pattern for positive symptomology without having the contrast of other ways that pattern can manifest in evolving trajectories. Okay, so here we're just showing the importance of capturing the direction of the flow in addition to just the position of these linear exemplars. So linear exemplar 4 and linear exemplar 8 sit in the same part of the embedding, which is in gray in the background on the bottom. EVO DFNC, linear exemplar 4 is vertical, linear exemplar 8 is more horizontal. Linear exemplar 8 also lifts to an EVO DFNC that's much more important in controls than in patients. And the two linear exemplars intersect, and you can see on the top row, circled are the two patterns, the part of the EVO DFNC, the points where they're intersecting. But both of them they're dynamically quite different, and that is really the point. And in one of them represents something that's distinctively seen in controls. And the other is a pattern that's observed without a significant group difference. This is one that's a property of being human, I guess. And they live right near each other in the embedding, and they are distinguished by their direction, which is to say, like the tangent to these curves is going in different directions and capturing different dynamics. So ongoing and future work will be in the following. We're working already quite a bit on stability and canonicity. Averaging multiple embeddings qualitatively changes the group level geometry, as we saw in an earlier slide. We're developing stabilization strategies that are not as geometrically disruptive. We're looking into similarity metrics to assess the similarity between large collections of high-dimensional evolving objects, such as EVO DFNCs. We're working on automated parameter optimization, and we're looking at something really fun that I didn't have time to present here, which is connectivity dynamics characterized through attractors and bases of attraction of flow along tangents to the embedded DFNC trajectories. So if you consider in the embedding, you take a given point, and you ask it to flow along the tangents to the embedding, along the gradients of the embedding that are determined only by curves associated with that are the embeddings of trajectories of healthy subjects versus only tangents to the trajectory to the embedded curves that are associated with schizophrenia patients. And what happens is these flows diverge. You start in the same place, but if you're moving along the group level trajectories associated with being healthy, you end up in a different place than if you move along the group level trajectories associated with being a patient. And moreover, they're very different. Even if you have a fixed attractor for both populations, the basins of attraction for those attractors are quite different. So this is fun and interesting, and something that we're working on at the moment. So I thank you for your attention, and if you're interested in learning more, please see our preprint on the bioRxiv multi-frame evolving dynamic functional connectivity.
Hello everyone, this is Jing from Beijing Normal University. I'm glad that you attend the symposium on the learning from high-dimensional neuroimaging data. Today my topic is multimodal fusion with reference, a key to better understand the linkage underlying brain behavior and disease risks, or subtypes. So the first question is, why should we doing those multimodal fusion? We know that the brain imaging technologies have enabled image acquisition at faster rates and increased resolution, also from multiple modalities. And all those international acceptable brain imaging datasets have facilitated the generation of big neuroimaging data. But the neuroimaging data itself is high-dimensional and often with limited sample sizes. So how to learning those useful information from this complementary information has been a crucial but challenging endeavor. So in these multimodal fusion methods, we aim to combine those different views into one and achieving an aim of 1 + 1 is bigger than 2. And usually those multimodal fusion techniques could be combined with individualized prediction technologies so that you could identify those highly replicable neuroimaging signatures. And we will show that in the below. So compared to those correlation in univariant modalities, the multimodal fusion aims to recover those covariant brain function and structure together in a covariant circuits or a network. And usually this kind of network is more robust to noisy conditions, therefore is more easily to be replicable. And we will show that in the next examples. So currently the most popular fusion methods include on this left panel, such as linked ICA, MCCA, joint ICA, and more recently our proposed MCCR plus GICA. However, many of these methods were purely blind, that is only based on imaging data but not incorporated those clinical measures. However, many clinicians and other cognitive neuroscientists would like to be interested in specific measures such as behavior, cognition, symptoms, or genetic variants. So how could we incorporate those measures of interest into the framework of joint decomposition between multiple modalities? That is what we will achieve for the multimodal fusion. And this is animation of the flowchart in those Fit toolbox. So usually for each modality, we will first extract features so that to get a more concise, informative presentation of each modality. And then we link them together into a single framework by maximizing different cost functions, such as covariation across modalities or joint independence. However, in those supervised learning, we could incorporate the measures of interest, such as symptoms, into this purely aligned framework, and thus we will optimize the cost function like this. And for more details, you could see the papers attached. So based on this framework, we usually have two kinds of research applications. The first is we could find the common and unique brain alterations between the groups you compare. And also, and more recently, people are more interested in this individualized prediction. So we could also doing those disease risk estimation or the prognosis prediction. So here I will give 2 examples to explain each of these aims. So the first is explore common and unique alterations in autism subtypes by symptom-guided 3-way theory. We know that the heterogeneity in autism spectrum disorder, for short ASD, present a substantial challenge to diagnosis and precision treatment. The whole ASD group can usually be divided into 3 subtypes, that is Asperger's, PDD-NOS, and autistic disorders, among which all those heterogeneity could exist in the biological etiologies, genetics, neurosystems, and many others. And in this study, we hypothesize that the heterogeneity of behavior symptoms is measured by these ADOS scores will provide another window into those abnormal multimodal covariant patterns that could potentially allowing for the comparison of similarities and distinctions among these subtypes. So here is the flowchart for this whole analysis. And we use the ADOS composite score as the reference to guide the FALF and gray matter fusion in the ABIDE-2 dataset. And based on those identified brain maps, we could build linear regression models to predict the corresponding symptoms in the ABIDE-1 cohort and to see if those identified brain signatures could be predictive in the specific subdomain scores and whether there are any common and unique impairments in those ASD subtypes. And here is the results. We could see that the dorsolateral prefrontal cortex and the superior and medial temporal gyrus are the common functional and structure covariant brain regions among ASD and all these subtypes. While the key difference among the 3 subtypes are the negative functional features that can be shown in the FALF, in the subcortical brain regions. Specifically, for the Asperger group, we found that the negative FALF was in putamen parahippocampus, and in the pervasive developmental disorders, we find that the negative FALF in the anterior cingulate cortex, while for the autistic subgroup, the thalamus amygdala caudate circuit showing the negative FALF. So we could see that all their subtype difference was reflected in the function instead of those structure regions. And more importantly, when we build based on those specific regions to predict the corresponding symptoms cross cohort, we could find that each subtype related patterns is only correlated with the ADOS subdomain scores, but not the others. Each subtype's unique multimodal patterns are correlated differently, while the social interaction is the common deficit. For the autistic group, it has more deficited social behavior impairment, while for the Asperger, which is more mild, it's only infected the social interaction. And here is a summary of all those findings. We could see that besides a common neural basis, with those core deficits involved in those ASD subtypes, each subtype was related to their own specific patterns, and this pattern is correlated with different ADOS domain scores. And more importantly, these features, the prediction model based on these features only predict the corresponding subtypes across cohorts. That it means from a byte 2 to a byte 1. So okay, to summarize, this study makes a key contribution to understanding the brain imaging heterogeneity in ASD under a multimodal fusion perspective and across cohort prediction framework. And considering those four following points, the first is the site was regressed out from the features we input for fusion, and we used partial correlation between the subtypes component and the ADOS, remaining still significant after regressing out the site. And third, the most frequently occurring voxels are similar with the original ASD pattern. And finally, the linkage between subtype components and ADOS are projecting from a byte 2 to a byte 1 remains significant. We could conclude that the side effect will not be a main factor affecting our current fusion results. One of the limitations of the study is that it's male-based, therefore it cannot be generalized to female or the more general ASD population. And finally, the identified core and unique subtype-related patterns may help us better understand the neural substrates involved in the behavior heterogeneity, particularly in the range of severity in autism. Such a method can be applied to many other disorders. And the second example is disease risk prediction based on imaging signatures identified from personality-guided 4-way MRI fusion. And here is the hypothesis flowchart, and we will show you how motivated us to do this analysis. So we know that adolescence is a critical developmental period in which personalities that extend to personal adulthood are established. And one of the largest behavior changes is novelty seeking, which access the preference for seeking novel experience and higher levels of rewarding stimulation. It is also a key behavior trait in adolescents that is of critical importance in transitioning from a dependent child into an independent adult. Therefore, the NS dysfunction can be related to several mental disorders, such as drinking, smoking, ADHD, depression, and schizophrenia. And there are previous studies have shown that the hypersensitivity of novelty seeking was related to some impairment in the reward processing symptoms. However, the specific associations and the longitudinal impact among NS, reward circuitry dysfunction, and risks for multiple disorders have remained explored. So therefore, could we use the personality-guided brain imaging signatures as a predictor to longitudinally predict those disease risks or even estimate their symptom severity when they become adults? So that is what we want to do in this study. We hope to understand the brain structure and functional components that are related to the NS and also play a role in forming personality differences in adolescent development. We aim to answer the 4 questions. The first is it possible to use high NS associated multimodal networks at age 14 to longitudinally predict the novelty seeking and 5 disease risk scores at the age 19 for the same subjects? And also, can this be applied to those unseen healthy subjects? The second is, could we predict the 5 corresponding symptoms spanning drinking, smoking, ADHD, MDD, and schizophrenia? And the third is, could we use this to further classify mental disorders? And finally, could we discover a general brain circuit that's linked with impairment of reward processing? So that is what we want to do in this whole design. Specifically, the normative thinking scores were used as a reference to guide the four-way multimodal fusion that include three tasks, three fMRI task contrast maps and gray matter volume to identify a set of multimodal imaging signatures. And the discovery dataset was selected from a subset of 14-year adolescents with the top 20 ranked high-NS scores. And based on those identified signatures, we will build models to longitudinally predict the seen and unseen subjects at age 19, and then further for the cross-cohort patient prediction and classification. And here, this is the subject sample size we have used. So at age 14 and 19, we used the imaging dataset that had about 1,100 subjects, but the discovery only contained 220 subjects with very high NS scores. And for those external validation, we have include drinking, smoking, ADHD, MDD, and schizophrenia at a different sample size. And here is the results. The most identified brain regions associated with high NS include the prefrontal cortex, striatum, amygdala, and hippocampus. Also, both longitudinal prediction and cross-cohort patient symptom prediction across 5 disorders were all works, which means the results were significant. And more importantly, the identified reward-related multimodal features could classify among multiple types of patient. The binary classification between all controls and patients is 84.4%. And for the 6-group, 5-group, and 3-group classification, the accuracy was 75, 79, and 87% respectively. More importantly, the AUC is 0.98 for classifying ADHD, MDD, and schizophrenia. In all cases, the AUC is higher than 0.91, showing promising for the clinical utility of the identified NS-associated biomarkers. And here is the summary figure. So basically, we could conclude that the high novelty in adolescent is associated with alterations in the reward processing that may implicate higher risk for subsequent development of drinking, smoking, ADHD, MDD, and schizophrenia. And more importantly, the identified high NS associated network may serve as a transdiagnostic neuroimaging biomarkers to predict disease severity as well as classify between patient groups. So the interim summary is that based on a rigorous longitudinal and transdiagnostic cross-validation, leveraging 6 independent datasets, our data-driven fusion and prediction analysis revealed that there is a degree of dependence among these 5 patients that are apparent partially shared deficits in addiction and psychiatric disorders. And all those may arise from a common NS-associated dysfunction. And note that for the external patient cohorts, we have only resting state fMRI data, while in the discovery, we actually have task fMRI data. So although they are resting and the task difference, we extract the same ROIs from the rest fMRI. And also, our results show excellent reproducible performance. Even the scanning parameters was different from those sites. And without specifically matching the imaging parameters, either in prediction or classification. And finally, collectively, the present study showed a contribution of personality, neural response, and structure covariation that capture a generalized multimodal rewarding circuit shared among 5 disorders and can provide important scaffoldings to support a framework for investigating neuromechanisms underlying addiction and psychosis in adolescents. And more importantly, besides the features we identified from multimodal fusion, we could further combine with different linear regression models as we have summarized in our recent biological psychiatry paper. And together with multimodal fusion and individualized prediction, we could get more reproducible imaging features that show some translational clinical impact. So to conclude, data-driven multimodal fusion is rapidly growing, but it is still underutilized. And the fusion methods could capture more views of brain aspects and covariation. More importantly, guided by other clinical measures of interest, we could identify those brain signatures more accurately by supervised learning, and which also show excellent replicability across cohorts, which could further instruct the longitudinal prediction of risk or treatment outcome prediction. The third, multimodal fusion involves more expertise and results interpretation involving a learning curve and multidisciplinary expertise is essential. And finally, emerging big data technologies and advanced machine learning methods, such as deep learning, could be combined with multimodal fusion, which show great promise for generalizable single-subject prediction. So finally, I'd like to thank all my students and postdocs, and here are other posters for you to interview in this OHBM conference. And also, I'd like to thank all my international and domestic collaborators and my funding support. Thank you so much.